So we are part of the H3 Africa consortium that you might have heard about yesterday. It's under the auspices of the African Academy of Sciences. And how the H3 Africa consortium came about was there was a white paper came out in 2011. And in this white paper, and these are slides that I took from the H3 Africa website, is that in the white paper, they highlighted the fact that Africa suffers a disproportionate burden of communicable disease. And that is, research is mainly focused on HIV, TB, and malaria. So they kind of dominate the research in Africa. But there's lots of emerging non-communicable diseases. They're just coming down the track. And genomics is really important. It's the key to understanding these diseases. But genomics and genetics in Africa is limited. There's limited collaboration among African scientists, and there's an African brain drain. Despite that, African populations have the greatest diversity, so there's a lot of opportunity there. Africa is uniquely placed to study gene-environment interactions, and the data that's coming out of Africa is needed in order to achieve a better understanding of ancestral genomes. So with that in mind, the Wellcome Trust and the NIH came together and set up H3 Africa. And its goal was to increase the number of African scientists that are working on genomics, establish collaborations across Africa, and increase infrastructure in genomics on the continent. So to do that, they put their hands in their pockets and they're funding collaborative centres. So these are the sort of large networks, of which Panagen is one. So I think they fund 13 collaborative centres. They also fund smaller research projects, a massive bioinformatics network across Africa, 3 biorepositories, and they've put quite a lot of money into ELSI, that's ethical, legal, and social issues. So this is the H3 Africa Consortium, about 500 members. And that's about $170 million of investment by the Wellcome Trust and the NIH, funding 48 different projects. So how did we get involved in this? Well, when the call came out in 2011, we put a letter of intent, a pre-application in, and the Wellcome Trust gave us £20,000 to have a network meeting in Uganda, so we could get all potential partners together and discuss a proposal. But we were already a network, and this is what we tried to say to the Wellcome Trust when they funded us, was that we already had lots of connections. So I worked a lot in East Africa, and my French collaborators worked in West Africa. So there's a lot of interconnections already. Not so much South-South connections or Francophone-Anglophone connections. So that's what we wanted to address. So we put in a full application in 2012. They interviewed 4 of us. It was very nerve-wracking, and then we were awarded it and we were funded in 2013. So that's 2 years after the first call came out. We managed to get a no-cost extension, and then we had to apply for refunding. Almost, only 3 years after we'd been awarded the grant money. So by 2016, we had to apply for refunding. So very quickly, we had to demonstrate that we can publish, that we can work together, that we've collected lots of samples. So we had to do that. We then went through the pre-application, the full application stage, 2017, got interviewed again. And then we were awarded it to start in April 2019. Obviously, and then so that was for 3 years. But of course, we've been massively hit by COVID. So it was bad timing in that regard. And so we're already thinking about how we can get refunding. The HD Africa scheme is going to end after this call. They're not going to renew anybody. So it was always going to be for a finite amount of time. It's part of the NIH's special fund. So it can only run for a certain amount of time. So they're not going to refund it. They've tried to come up with another scheme that's kind of quite similar. So we're exploring that at the moment. So that's about how we got funded, but what is Japanogen all about? Why did we want the money to do this? Well, our main objective is to identify the human genetic components that determine susceptibility to trypanosomiasis. So in the first round, it was trypanosomiasis. When we got renewed, it's Japanogen Plus, because we introduced schistosomiasis into the scheme as well, to show that we're expanding. So we are working in countries where trypanosomiasis and schistosomiasis are endemic. And we're looking at the diversity of infectious outcomes. So either once you're challenged, you can either have no infection, or once you get an infection, you can have a range of disease outcomes from asymptomatic carriers, mild disease, severe disease, death, high worm burden, low worm burden. For both diseases, there's a range of disease outcomes. So we're exploiting that to look at the genetics of both of these diseases. So we're doing two different approaches. For trypanosomiasis, it's a genome-wide association study. For schistosomiasis, we're doing a family-based association study. Just because we have access to children and their parents, and that's a much stronger approach. But underpinning both of these is capacity building. Capacity building is a huge part of Japanogen and a huge part of HD Africa Consortium. So by capacity building, we mean we are training a lot of students, PhD students and postdocs in genetics, genomics, even ethics. We've got a PhD in ethics, public engagement. We've got our biobanking as well. We provide on-the-job training with an expert bioinformatician. And we've also generated resources, sort of bioinformatics, genetics data, but also our biobank of samples. And we hope to be building a research culture, hopefully that is sustainable and African-led. And we're trying to build a cohort. That's why I've got a picture here of us all wearing our Japanogen yellow t-shirts to give us a sense of community. In terms of community engagement is also a big part of Japanogen. We work with the national control programs and community leaders and healthcare providers. We're trying really hard to get informed consent. And so that involves real working closely with the population. So doing things like putting on theatre plays, documentaries on national television, museum exhibitions at local schools, that kind of thing, to ensure that people are understanding what we're about. And Japanogen has grown from having 9 African PIs to 11. We've got more PhD students, we've got more postdocs. And importantly, we've got on our ISAB board, the Scientific Advisory Board, we've got more African advisors. And so I'm just focused on trypanosomes because schistosomiasis projects is really in its infancy still. So for trypanosomiasis, we were aiming to diagnose and enroll greater than 3,000 cases, carriers and controls and create a biobank of that. We also wanted to sequence at least 300 individuals just to get the local SNPs relevant to our populations that can be then used in the African SNP chip, the H3 Africa SNP chip. So all our data was used to inform on the SNP chip, and we've used that H3 Africa SNP chip for our GWAS studies. So I'll just cut to the chase. So we've managed to carry out a genome-wide association study using the H3 Africa SNP chip. And this was from our discovery cohort. So we have Manhattan plots for infected versus controls, cases versus carriers, and carriers versus controls. And we've got some hits. And then we then took those hits and looked in a validation cohort, so a smaller number of samples, different samples though, and they still were significant. And so we're taking these further now for validation experimentally. So the key to all of this we're working on in Trypanogen is accurate diagnosis and phenotyping. And that kind of links on to the next thing that Martin wanted me to talk about, which was diagnosis. So for Trypanogen, for identifying people infected with rhodesiense, it is pretty easy. You're just looking for trypanosomes in a blood smear. Or you could use a mini ion exchange column to concentrate the parasites. But really, your parasiteme is normally quite good, and so you can see them under a microscope. So if you're positive by microscopy, you then get a lumbar puncture to determine if you've got parasites in your CNS, or if they're just limited to the blood compartment. And if you don't have any trypanosomes in your blood, you're considered a control. That's fairly straightforward for rhodesiense. But what about gambiense? That's not so straightforward. And that's because recently we've discovered that there are such things as asymptomatic carriers, and they act as a reservoir for the disease. So these asymptomatic carriers are not diagnosed, they're not treated, and they're not included in prevalence data. So for diagnosing gambiense, typically the parasitemia is really low. So we use firstly a screening process, a serology test, which is not really available for rhodesiense. It's called the CAT test. And if you're serology positive, we then take your blood, put it down a mini ion exchange column, do a lymph node aspirate, also take that, and then look under the microscope to see if you've got parasites in the blood or in the lymph nodes. Then if you're positive, you're then staged with a lumbar puncture. But what happens if you're negative? So you're seropositive, but you haven't got any parasites in your blood. These people could be potential carriers. And so what we've done is follow these people up for at least 2 years. And if they're still positive by serology, and for a second serology test called the trypanolysis test, then we consider them as carriers. So could these asymptomatic individuals contribute to disease transmission? And where are these parasites in these individuals, in these asymptomatic carriers? So to address the first question, we collaborated with Katie Atkins, a modeler, who showed that if you just take the number of cases alone, there's not enough cases to sustain a focus of disease. So the R number, R naught, is about 0.8. If you take the asymptomatics on their own, they're not enough to maintain a disease focus. But together, the R naught is greater than 1. So you So asymptomatics, human asymptomatic carriers plus cases is enough to maintain the disease without even thinking about an animal reservoir for Gambiense. So how can these asymptomatic individuals contribute to transmission if they have no detectable parasites in their blood? Well, for that, we have to understand how the tsetse flies take a blood meal. So they're pool feeders, and this is a picture of, can you see? A pointer. Oh, it doesn't matter. Of the proboscis of the tsetse fly. And it's got rasps at the bottom and teeth down its edges. So it really mashes up the skin as the fly takes a blood meal. And then it feeds on the pool of lymph and interstitial fluid and blood that collects. So the question was, could the parasites be hiding in these other fluids, that is in the skin? And the short answer is yes. And so we looked in the skin of infected mice and stained them with an antibody for trypanosomes, and they're stained brown here in this picture. And you can see trypanosomes at all depths of the skin. And if you use bioluminescence to look at infected mice, what we found is that most of the bioluminescent signal is actually coming from the skin. So there's a lot of parasites in the skin. Are they alive? Well, we used 2-photon microscopy to demonstrate that these parasites in the skin are all happy and wiggling about. Here they are here. And this white thing here is the blood vessels. So you can see that the parasites in red are outside the blood vessels. So the question is, do these skin-dwelling parasites contribute to transmission? And to address this, this is work done by my collaborator, Bruce Rotoro, who took, as you'll know, that the parasitemia in a mouse fluctuates. So sometimes there are lots of parasites in the blood and sometimes there are very few or not detectable. So he took a mouse that had no detectable blood parasites, but did have skin parasites, and fed tsetse flies on an area where there was no bioluminescence and an area where there was bioluminescence. And none of the flies got infected if there was no bioluminescence there. And about a third of the flies got infected if they were fed on an area where there was bioluminescence. He then took a mouse that was in the peak of parasitemia, so there was lots of blood parasites as well as skin parasites, fed tsetse flies on an area where there was no bioluminescence. 2/3 of them got infected, so they're getting infected from the blood. But to get 100% transmission efficiency, you need both skin and blood-dwelling parasites to be there. So that looked like skin-dwelling parasites are required for maximum transmission efficiency, even in the absence of blood parasites. That was in the mouse model. So to see if that was true in humans, we went to Guinea with my collaborators to Forogueria, and this was the Great Trip. And so we were looking for trypanosomes in the skin of individuals. And we tested 5,500 people for serology. And of those, what we noticed, first of all, is that the people that were stage 1 or stage 2 disease or the seropositives, quite a high proportion of them had dermatological symptoms. That is rashes or itching or some sort of lesions. And that these dermatological symptoms disappeared when they were treated. So before treatment, we took skin biopsies of these people and did touch preps, and then took the samples back to Glasgow to do immunohistochemistry. And this is just an example of a positive sample So it's again stained with a specific antibody to trypanosomes. That's the brown stain. And these are zoomed in pictures, times 20, and there you go at times 60. You can see the brown trypanosomes within the skin. There's another pictures there. And from all the seropositive, okay, it's a small sample size. We've got seropositive cases, stage 1 and stage 2. All of them were positive by immunohistochemistry for trypanosomes. So it looks like trypanosomes accumulate in the skin during a normal infection, and that creates an anatomical reservoir. And the skin-dwelling parasites can contribute to transmission, even in the absence of blood parasites. So that's in the mouse model. And we can detect trypanosomes in the skin of carriers and cases in humans. So what are the implications for this finding? Well, we don't really know what the true prevalence of the disease is, after all. We've been looking in the wrong place all this time. We've been looking in the blood when we should have been looking in the skin as well. And now there's mounting pressure to close control programs, and that puts the elimination at risk of re-emergence, especially if carriers are not treated and are left to transmit the disease. So the next thing is, can we improve diagnosis to include these individuals with skin-dwelling parasites? Obviously, it's not a normal part of the diagnosis to include skin biopsies. So how do we detect dermal trypanosomes? Well, we could do skin biopsies, although the touch preps, it's quite quick, it's quite cheap, it requires a trained reader to do it, and it's quite labor intensive. We could try and culture trypanosomes from skin biopsies. And so my collaborators are trying this, taking a skin biopsy and putting it in media called kiwi, and it transforms the parasites. And hopefully after a few weeks, you will see trypanosomes in the culture media. Obviously, that takes a long time, and there are contamination issues, and you require culture facilities. So it's not really an easy thing to try. You could do molecular biology, so do PCR. We've got lots of PCR targets for trypanosomes that are specific for Gamiense that we could try. You'd need to make DNA in the field, but that's okay, that's feasible. And it's very sensitive. There are contamination issues, and you do require PCR machinery, etc. The other options are xenodiagnosis. And that's not really an option, because we've been telling people for quite a long time that tsetse flies transmit disease. You can't then put tsetse flies on them. That would not go down well. Other methods would be for scanning the skin to see if their trypanosomes are there. And one of the methods that my collaborator, Bruce Rottero, is trying is this chronometer. I don't know how you pronounce that. It measures capacitance. Can't even say that either. It's basically the hydration of the skin, and it's very quick and very easy to use. And he's got some data that suggests that as somebody is treated, the capacitance or the hydration of the skin decreases. So that suggests it's a good marker for being infected. But of course, it's indirect, and it is prone to other factors such as the ambient temperature. But it is very easy and quite cheap. But the method that we've been focusing on is using Raman as a method to detect trypanosomes. So what is Raman spectroscopy? Well, it involves shining a laser on a sample and detecting the scattered light. And most of the light that comes back is not changed. but a very small percentage of the light is shifted, and that's a Raman shift. And you can plot the shift in light on a spectrum like this. And that spectrum is unique to whatever you shone the light on in the first place. So we have used one of the peaks of the spectrum, so it's 2934 wavelengths. We've used that in a Raman microscope, and you can see that it lights up the trypanosomes very well. But that's a trypanosome on a glass slide. How does it work distinguishing infected and uninfected skin from a mouse, for example? So that's what these spectra are at the bottom here. Can you see my cursor when I move it across? Yeah. The blue is uninfected, and the red are infected. So you can see there are some differences here in the spectra. And if you do a PCA plot, the infected mice are clustered together and away from the uninfected skin. So that was looking quite promising. And if you leave the mice for longer, so you get longer infection after 3 weeks, you get an even bigger difference from uninfected to infected. And these are two different trypanosome strains. One goes into the skin more readily than the other. So you can see the difference here. Some peaks don't change. So this is an invariant one. And so what you can do is take the ratio of an invariant versus a variant peak and come up with a ratio that can used as a marker for identifying, for saying you've got trypanosomes there. This is these spectra are from a more detailed time course of infection, and as the time goes on, the peaks that are different between infected and uninfected increase. So if you take the peak ratio of, say, that one with that one, and plot that, you can see that the peak ratio increases as the infection progresses. So this is, and what I should have said was the Raman can detect the trypanosomes in the skin 4 days before you can detect it by qPCR. So the Raman looks like it could be a promising tool. It's possible to adapt it for a little handheld instrument. We're using a portable instrument at the moment, but you can get smaller ones. We're testing more patients in the field at the moment and comparing that with skin biopsies. It can also be applied to dry blood spots, which would be quite useful. And it can be adapted to use for blood, for the whole blood, by using nanoparticles that can enhance the signal. And this is just an example of the nanoparticles that you can use to enhance the signal. This is control blood and this is infected blood. These have been offset so you can see the difference in them when it's not so clear when you overlay them. So Raman tool is non-invasive unless you're using a finger prick, but even then, that's minimally invasive. There are no running costs unless you're using the nanoparticles. It's specific, we hope. It's very sensitive and can be applicable to other vector-borne diseases. And it has been used in, it's been tested for Chagas disease. And that looks really promising. And it is being used at the moment, or being developed for other infectious diseases such as malaria. Malaria is particularly good because the infected red blood cells have a lot of hemozoin, which has got a really good Raman signal. So that's looking very promising. So these are perhaps things we can try. So I'm coming to the end of my talk. I've given you a whistle-stop tour of Trypanogen and my consortium and how we're managing. I don't know if I have touched on how we're managing COVID, but we're struggling. But I focus on the disease approach, the diagnostic approaches that we're adopting. So I'd just like to finish by acknowledging my collaborators around Glasgow and also the Trypanogen network.
Professor Jackie Cook, she's an associate professor in malaria epidemiology at the London School of Hygiene and Tropical Medicine. She uses, sorry about the tweeting in the background, that's my Twitter stream. She focuses on using serology to look at malaria transmission. She's done a lot of work in Cambodia, but also across sub-Saharan Africa. And she's been based in Zanzibar previously to try and focus on various different surveillance methods as applied to malaria elimination. And she's also more recently worked substantially on vector control. So she's got all sorts of expertise and experiences in the global south, but on the other side of the Atlantic, which might be super important to try and understand how we can get a grip on what's going on in Venezuela, especially in terms of trying to understand what the burden of malaria truly is at the moment. Take it away, Jackie. Thank you.
Thank you, Mark. Thank you. Thanks, Martin. Thank you for the introduction, and thank you very much for inviting me to speak. So I'm at the MRC International Statistics and Epidemiology Group at the London School of Hygiene and Tropical Medicine. And as Martin said, I've kind of got a few different hats when it comes to vector-borne diseases. So initially, my work was primarily focused on using serology to look at malaria surveillance. And more recently, I've been looking at evaluating vector control interventions in big trials. So looking at new nets and looking at Eve tubes and house screening is a trial we recently finished in Côte d'Ivoire. So I'm very happy to be invited. And if you have any questions about any of those areas of expertise, I'd be really happy to talk to you about them. And hopefully they might come in useful in the brainstorming session. But today I'm going to talk specifically about the serosurveillance of vector-borne diseases. And there's going to be a big lean towards malaria because that's mainly what I've been working on, but I have included a few slides for other vector-borne diseases as well. And just a hat tip to Chris Drakely's group at the London School of Hygiene and Tropical Medicine. So that's the group that I used to be in, and they are continuing this work and still do a lot of work in this area. And also a nod towards Kevin Tete, who is the person who makes all the antigens which make this work possible. So the reason that we can use serology as a proxy for transmission is that humans develop antibodies to infections following exposure. So that can be a parasitic infection or a virus, for instance, but those antibodies don't always result in effective immunity. So you might still remain infected. But the evidence of having antibodies in your system shows that you've been exposed at some point in your life. And we can look at the population level, at the seroprevalence in a population to have a sort of proxy of how much infection there is in that community. And so you might be more familiar with serology in the past year because it's been used for COVID, for instance. So to look at how many people in a population have been exposed to the infection. So when transmission of a disease is stable over time in a population, you would expect many more adults to have antibodies than children. And that's just because they will have had many more opportunities to have been exposed. And the caveat to that is if it's an infection that only induces very short-term antibodies. So if people are making antibodies but then losing them very quickly, then you won't see this pattern of adults having more antibodies. But we can still use that data to say something else about the infection. So this is a diagram just to show how this works in terms of malaria, and it's also quite similar for other vector-borne diseases as well. So when you're exposed to an infected mosquito, you make an antibody response and you may end up remaining infected or you may clear your infection. Those antibodies may or may not be responsible for that. And when you're exposed to many infected mosquitoes, you develop proportionally more antibody response. And again, you may still remain infected or uninfected. And so this top panel is potentially representative of a low transmission setting where you're not exposed to infected mosquitoes very regularly, and therefore the level of antibodies in the population is quite low. in comparison to the second panel, where people are exposed to infected mosquitoes quite regularly, and therefore there's just a lot more antibody in the population. Or you could think of that top panel as representative of a child who may live in a very high transmission setting, but they've only just started on that journey of being exposed to infected mosquitoes, and therefore they have a low level of antibodies, compared to the bottom panel, which would represent an adult who is being constantly exposed and has over their whole lifetime, and therefore will likely have a lot more antibodies in their blood. And so it's this phenomenon that we use to model the data to be able to use serology as a proxy for transmission. So if, for instance, you went into a village and you tested everyone in a village for, tested their blood to see if they had antibodies to the malaria parasites, you might expect to get a result of something that looks a bit like this. So here on the x-axis, we've got age, and then we've got the proportion seropositive on the y-axis. And as you can see, that increases with age. And that is just an artifact of the fact that adults are going to have had a whole lifetime of exposure. So you're just going to get more adults who've been exposed than children. And so we can fit this data with a simple model to allow for the rate of seroconversion and seroreversion. And that gives us a lambda, which is the seroconversion rate. And so this lambda suggests that 7.5% of this population is seroconverting every year. And essentially, the reason we want to get a number out of this is just so we can compare between different settings. So we know that it's going to be very much related to the age structure of the community, but by fitting this model, we can generate a seroconversion rate for that population. And then we can then start to look at different seroconversion rates within, between countries and then also within countries to try and get an idea of the transmission intensity. So these are some examples from some of the settings that we've done work in previously. So on the left here, we've got Vanuatu, which is a very low transmission setting. In these graphs, the red triangles represent the actual data points and the blue lines represent the model that we've fitted to the data. And you can see what we're really interested in is that steepness of the curve. So that represents the rate of the seroconversion in the population. And so when we're looking at this low transmission setting, we have quite a flat curve, a very slow seroconversion rate, quite a small lambda. And that increases as we move across into a moderate transmission setting. And then when we get into a high transmission setting, we're looking at quite a steep curve, and people are actually getting antibodies quite quickly in that particular population. So this is looking at different settings with very different transmission intensities. But the idea behind using this kind of method is to make it useful for control programs, for instance. So if you were able to get these kind of curves for different districts in a country, so thinking of Venezuela, then potentially you would know where you would want to target your interventions. If you don't have enough interventions to give them out to the whole country, you will be able to look and see where is the most transmission occurring, where is the place where we're going to get the most benefit from these interventions in terms of reducing transmission. So the way that this way of analysing the data came about was through Chris Drakely, who's based at LSHTM. And so he was based in Tanzania, and back in 2005, together with colleagues in Tanzania, they collected samples from the Usambara Mountains, which are in northern Tanzania. And so in each of these villages, so each box represents a village, they were collecting serological samples and measuring antibody responses in those samples. So on the x-axis, again, we've got age, and on the y-axis, we've got proportion seropositive. And so starting at the very top of the mountain, there's very little malaria transmission at high altitude, just because the temperatures are very cold. And so these villages are at the very highest altitude point, and you can see that we've got very flat curves, quite small lambdas, not very much transmission going on there. And then as we move further down the mountain and we lower the altitude, that's when we start to see these curves getting a bit steeper. The lambdas are starting to come up until we're at ground level, sea level, ground level, where we can see that we've got the highest transmission going on. So we've got the steepest curves and the highest lambdas. And so this is when the method was still being validated. And so in each of these villages, they also made an estimate of entomological inoculation rate, which is the mean number of infected bites that people are receiving over a year. And so this was considered the gold standard for estimating malaria transmission. And then they compared the results that they had for antibody responses or seroconversion rate for 3 different antigens on the malaria parasite to entomological inoculation rate. And they saw that they correlated quite well. So that was suggestive that using these serological measures and this seroconversion rate was quite a good proxy for transmission in this setting. But why do we need another way to measure transmission? And I think in Annette's talk, it was quite interesting to hear how it is quite difficult to diagnose diseases still. And we don't necessarily have that problem in malaria, but we do have other issues in terms of not being able to detect all the infections that are present. So for instance, we can look at passive data collection at health clinics, so waiting for people to come in and to be diagnosed with malaria. But we know that is going to miss a substantial amount of infections, and that's true for malaria, and it's definitely true for other vector-borne diseases as well. There's a large proportion of them that are asymptomatic, so you're not going to ever see those clinical cases coming forward. We also know there's going to be a proportion of the population who aren't going to be using those health clinics, and so they will go unrecorded as well. And often that might be people who are potentially more at risk of vector-borne diseases, such as migrant communities who just aren't integrated into the communities to use those facilities. Or, for instance, in Cambodia, it would be forest workers. So they're at particular risk through their work in the forest, but they're often not reporting to their health facilities. And I guess it might be the same in Venezuela with your gold mines as well. We also know that some vector-borne diseases are very difficult or expensive to diagnose. So some require molecular diagnosis, whereas the serology technique is quite cheap. Entomological measures can be quite labour intensive. So we heard a bit yesterday from Roberto about doing AD surveillance, and it is a labour intensive method, and you can end up finding very few mosquitoes. And if you've got very few mosquitoes, it's quite difficult to be able to tell, A, if your interventions have had any impact, or B, if you've got any differences in transmission intensity, for instance, between different settings. And also vector-borne diseases are seasonal because they come along with their vectors, which means that if you're doing a cross-sectional survey, you need to time your surveys quite carefully. So serology has some advantages over some of the methods that are used already. So in particular, it's quite sensitive, particularly at low transmission levels. And that's because antibodies just hang around for longer than the infections would. This is particularly for malaria. So it means in a very low transmission setting where you've got very few infected mosquitoes to find, you can only find a few parasites within people, you will still be able to find quite a lot of people who have antibodies, so representative of previous exposure. And because those antibodies last a relatively longer time, and different antibodies last different amounts of time, but they are relatively less affected by seasonality than things like infection prevalence. And within one sero survey, you can simultaneously give an estimate of current transmission, and you can try and evaluate historical transmission as well. So if there's been a change in transmission, you can pick that all up in one survey. And I'll give some examples of that with malaria and also with other diseases in the talk. Measuring antibodies is quite simple. So we tend to use filter paper samples, which are easy to collect and to store. It's quite high throughput and relatively cheap if you use ELISA. So that only requires quite basic lab facilities. If you do have a bit more money to put behind it, then you can use multiplex technology, which means you can detect antibodies to multiple antigens simultaneously, which vastly increases the amount of data that you can get. And you can look at different diseases at the same time. There are, of course, some caveats also to serology data. So I'm conscious this is a diagnostic session, and at least for malaria, we definitely can't use serology for diagnostic purposes, but we are looking at short-term markers, so antigens that induce very short-term antibodies, which are then representative of very recent exposure. And that's quite interesting to look at, particularly in elimination settings, or to look at the impact of interventions, for instance. Also, as I've said before, those antibodies don't necessarily mean, they don't equate to immunity. They don't mean anyone is protected necessarily. We're just using them as markers of exposure. And the choice of antigen that you use is really key. And I think that's something that we can exploit. And I mentioned Kevin Tete, who is in Chris Drakely's group, and he has made a whole array of, or a whole range of antigens for malaria, but also for other things. And we can choose the antigens based on their immunogenicity. So for instance, in a high transmission setting, you might want to use an antigen that isn't very immunogenic, so you don't get like topped out with the antibodies, and you can still differentiate between settings. Whereas in a low transmission setting, you might choose an antigen that is very immunogenic because you want to detect any antibodies that are there. You want to pick antigens that have limited polymorphism between strains of parasites, so that you don't only measure antibodies, for instance, in one setting, but that you can't measure them in other settings. And ideally, you want your antigens to be species specific. So in malaria, we've got 5 human malarias, and we have antigens that are specific for all of those. Obviously, there is always going to be some element of cross-reactivity, so you have to be aware of that. But ideally, you would get antigens that are specific for the individual species. So there's a number of criteria that we'd like to fulfill with any test that we use to antibodies. And I'm just going to talk about the 2 methods which I think are the most widely used now. And that's ELISA and Luminex. And they both have their own advantages, and they come with very different price tags. So I'll talk about ELISA first, because I think this is probably the most widespread method that's used. And when I was doing my PhD, I did tens of thousands of ELISAs. So ELISAs are relatively cheap. Cheap to do, so it requires only quite basic lab equipment. It's quite high throughput. So this example in this picture is a 96 well plate where we were testing 40 samples in duplicate. So you can get through quite a lot of samples quite quickly. Again, we tend to use blood eluted from filter papers, so that's quite easy to do. And you can test a few antigens at a time. So in general, you would just coat the plate with 1 antigen. But if you're looking, for instance, in an elimination setting and you want to just see if there's antibodies to anything malaria related, you might want to pool those antigens in the plate, and you can see if there's antibodies to any of them at the same time. Luminex is really the extension to that, the more expensive extension to that. So these machines range from about 40,000 UK pounds up to around 120,000 UK pounds. So it can be quite expensive. But you can simultaneously test many, many antigens at the same time. And so you can look at different diseases. You can have lots of antigens from the same disease. You can look at different subtypes of antibodies. You can look at IgG, IgM. So you basically really dramatically increase the amount of data that you are going to get out of that. But obviously, it does require having one of these Luminex machines, which are quite expensive. And then I just wanted to briefly mention a rapid test, which isn't something that is available now for serology, but it is something that is in development within Chris's group, but also within groups at WeHi and within in Paris as well, where they are looking at developing a rapid test to measure antibodies, one of these short-term markers, so they've got evidence for very recent exposure. And the idea for that is that it might be something that they can use as a kind of test and treat strategy. So if you have these antibodies, it means you've been infected within the last month, for instance, and then you might want to give people prophylaxis within that immediate area. So that's something that's still in development currently. And I've touched upon this a little bit already, but thinking about the different antigens that you might use. So within the malaria parasites, some of these antigens are quite well characterized, and we know that some are more immunogenic than others, and some elicit longer term antibodies than others. So some of these antibodies will last for decades. Others can last for just a year, for instance, for these long-term markers. And that kind of information is really useful for risk stratification. So I mentioned the idea of thinking about where you're going to put your interventions, and you can stratify places based on the seroconversion rate in those different areas and get an idea of the transmission that's going on in those places. You can also use them to evaluate the impact of interventions as well. So to see if you have a reduction in seroconversion rate after you've put interventions in place. We're also looking at these short-term markers, which elicit antibodies that don't stick around for very long. And that's really useful for looking at immediate impact. So you would expect all of these antibodies, if you totally got rid of malaria in a community, you would expect no one to have these antibodies within, say, a few months. And so that's a way of evaluating that. And as I mentioned, that's coming into line with that rapid test and thinking about using it as a test and treat strategy. And it's also useful for confirming elimination. And as I've touched on a little bit, you can use combinations of antigens. So you don't just look at one thing at a time. You can look at lots of different antigens from the same parasite or from different parasites. And then within the Luminex technology, you can look at different species and different diseases simultaneously, all with that same filter paper sample that you've collected. So this is a diagram from a Brian Greenhouse paper, just looking at some of the use case scenarios for serology. And you'll notice the range of transmission intensity here is from low to 0, which I don't think Venezuela necessarily falls under at the moment. But just to highlight that these top 2 surveillance methods, the stratification of risk and the measure changes due to interventions, that is very valid to do at high and moderate transmission as well. So I think that's where this kind of method would come in useful within Venezuela. So I'm just going to now show a little bit of data demonstrating some of those use case scenarios, and then I'll show a bit of data from serology for other diseases as well. So this is some data just demonstrating the use of serology for stratification of risk. And some of you might be aware of the Malaria Atlas project, which used to be based out of Oxford. And they make these amazing maps looking at parasite rate across the world. And we have these very impressive risk maps using parasite rate. And we wanted to see what they would come up with if they used seroprevalence as their underlying measurement. And so this is based on data which was published by Ruth Ashton in the Malaria Journal from serological data from school surveys in Oromia state in Ethiopia. And so on the right here, you see the typical Malaria Atlas project map of that state, where the bright colours represent areas where there is higher parasite rates, so higher transmission going on. And on the left, we've got the seroprevalence map. And so this data has been extrapolated from the schools data. And what you can see is those bright spots are really correlating quite nicely with the data from the parasite prevalence. But actually, we seem to have more granularity with the seroprevalence map. So we can see other areas which are potentially higher risk for transmission in the serology map than we can see just in the parasite prevalence map. And that really highlights the extra sensitivity that you get using serological data. And so these kind of maps, they don't have to be done using the more complicated methods that the Malaria Atlas project use, but the kind of maps where you look at seroprevalence over a district or between districts can be really useful just to know where your hotspots of transmission are. And they can also be used similarly with looking for population at risk. So if you have a particular demographic that you think is being more at risk of malaria, then you can particularly aim your surveys at that demographic to try and look at that. And this is some data looking at measuring changes in transmission intensity. So this is following some interventions. This is data from Bioko Island in Equatorial Guinea. And they started very intensive interventions back in 2004. So they were doing indoor residual spraying every 6 months, and they did mass distribution of bed nets. And they were able to reduce malaria, and that project is ongoing today. We were there in 2008 and collected serological samples. So that was 4 years after the interventions began. And we wanted to see if the serology could detect the change in transmission so soon after the interventions had been put into place. And so these are just 3 areas of the island: the northwest, Malabo, and the northeast. And this is one of those seroprevalence curves, so age versus probability positive. And you can see that we've got quite a steep curve representing quite high transmission and quite a high lambda or seroconversion rate there. And then when we look in the other 2 areas, what you'll notice is that there is a slight kink in these curves at the low ages. And so that's suggestive that those young children are undergoing a lower rate of transmission than their older counterparts were. So suggesting that transmission has changed within recent years. And so when this is a situation, we're able to fit 2 lambdas so that we can predict a lambda for the youngest age group, so a lambda 1, and predict a lambda for the historical transmission, which is the lambda 2. So here we were able to show that transmission had reduced, was over 3 times lower in the Malabo area, and was over 5 times lower in the northeast area, all based on this serological data. But we realized that we were probably losing some data by converting them just into negative and positive. Whenever you turn anything into a binary variable, you lose some information. So we also looked at the raw data, or the antibody titers, the strength of the response. And so this is a plot of all the different areas on Bioko Island again. And these are different age groups along the x-axis. And we're looking at the median antibody response on the y-axis. And so what you can see is that we have very low median responses in anyone under 5 years old in all of those areas. And we see quite steep increases in the age groups that are older than that. And we also see that in the northwest area. So the northwest area was the one that I showed you previously, which didn't seem to show any change. And so this is really just to highlight that even if you don't see a drop in seroprevalence, if you look more deeply into the data, that's when you start to see the impact of your interventions on the actual strength of antibody responses. And so this again is using the strength of antibody responses to do some analysis. So this is some work from mine and Chris's PhD student, Henry Surendra, who is based in Indonesia. And the idea behind this work was to include a surveillance system which was very little work for the actual health facility. So it was supposed to be a convenient sampling, I guess is the way to phrase it. So Henry was working in Kulon Progo district, which is a very low transmission setting. And what he did was he asked everyone who attended the health facilities to give a filter paper sample. So it didn't matter what they were there for, they were just asked to give a filter paper sample. And he also asked them to identify on a map whereabouts they lived. So he was able to get GPS points for where they lived in the setting. So it was minimal work. Obviously, he also did some QC of that kind of work as well. But overall, it was minimal work for the actual health facilities. And so what he did is he ran Luminex on the filter paper samples. So he got all of these different serological results for those filter paper samples. And on these maps, the dots represent age-adjusted antibody responses. So wherever you see a dot, that's a person, an antibody response. And where the dots are red, that means that's higher than expected antibody responses. And alongside the serology work, the clinics obviously continued their passive detection of malaria cases. And so during the year that this study was ongoing, there was actually an outbreak of Vivax. And Henry was able to incorporate that into his analysis. So what he did is he was looking at the serological data, and these black circles on the maps, I'll just highlight them there, are serological hotspots, so clusters of higher than expected antibody responses. And so you can see that some of those clusters are very consistent throughout the year. So these maps represent Q1, 2, 3, and 4. So those clusters aren't particularly moving throughout the year. And if you look on this second map, you'll see that those black triangles, they represent the Vivax cases that were detected. So the actual clinical cases, and they pop up in one of those serological clusters. So what we hypothesize is that actually, when you're getting outbreaks in these low transmission settings, they're going to pop up, or they're most likely to pop up in areas where historical exposure was higher, because that's where your mosquitoes are. So if, for instance, the program had used these serological hotspots as an idea to where to target their interventions, potentially that could prevent some future outbreaks. So I'm just going to briefly change tack and then just show you how this can also be used for other diseases, because I think that's something that's also exciting, particularly in the Venezuelan context, to get an idea of transmission of other diseases simultaneously, just potentially using this one sample that you collect, this one filter paper sample. So this is actually data from Venezuela, although quite old data from Venezuela. And I used to use this in a serology lecture, so I thought I'd bring it up just because it is in Venezuela. And you'll see that these plots look quite similar to the things that I've been showing for malaria. So here again, we've got age on the x-axis and proportion seropositive on the y-axis. And you can see that it increases with age. And these were surveys that were done every year between 1993 and 1999. And then the authors then fitted a force of infection model. So the black line is a year-on-year force of infection model where they estimate what the transmission was in that individual year. And because you've got all of these age groups, it means that you can actually estimate very historical transmission as well. So you can plot that. So that's what they did, and they plotted transmission, Chagas disease transmission all the way back to 1940s. And here on this figure, they've also plotted where some of the interventions started. So the green line represents when they started house-based spraying. The blue line is when they started a house improvement program, and the red line is when the national control program started. So you can see that there has been a massive drop in Chagas disease over the decades. And they've also plotted this on a map, and you can see that that has been uniform across the region that they were looking at. But that even in the, well, I say most recent, it's the 90s, so it's not very recent now. But in the most recent surveys, there was still seroprevalence there, suggesting that there is still ongoing transmission. So this is a way where that kind of data could be incorporated into serological assays. And so there's also similar ways that you can look at dengue transmission with this as well. So this is just an example from Singapore. So in Singapore, they implemented really extensive vector control in the late 60s, and they really decimated their population of Aedes aegypti, though they do still have outbreaks of dengue now. And they had these big population surveys looking at dengue-specific IgG. And again, these graphs look similar to the malaria ones, where you see that increase with age of seroprevalence. And they fitted a year-on-year model to estimate the force of infection. So that's represented by the orange line. And you can see that you get this kind of step through the data between the 1983 and the 1992 data, where you're getting this tailing off of people who are seropositive in that younger age group. And then by this 1993 graph, if you look just at people under 15, which is what this graph shows, you see there's very little seropositivity there, suggesting very few of them have been exposed to dengue. So just to summarize, so sero measures are a useful addition to other surveillance methods. They can provide a measure of exposure, but they can also look at changes over time. They correlate well, especially for malaria, that's where we have the most data, but they correlate well with other measures of transmission. And you can also say something about disease dynamics as well. The technology that we use to measure antibodies is quite simple. It's transferable. This doesn't have to be done in London. It can be done in any lab around the world. It can be really cheap doing ELISA. It's very easy to train people to do ELISAs. But if you've got a Luminex, that's even better, because then you can look at lots of antigens, lots of diseases. It's adaptable to different species. So within the Drakely group at the school, we've got Kevin Tete, who makes a lot of these antigens himself, but there are also commercial antigens that can be bought. And so you can look at lots of different species. I've listed a few there. You can also measure antibody responses to vector saliva, so to see whether you've actually been bitten by the vectors as well. And Kevin is also currently looking at incorporating SARS-CoV-2 antigens as well. So potentially that would be something to include at the same time. I've just put my email address there just in case anyone wants to contact me, but thank you very much for listening. And again, thank you to Chris Drakely's group, and especially to Kevin, who does all the antigen development and production for the group.
It's a great pleasure to introduce to you Dr. Alberto Paniz. Alberto did his medical training in Venezuela and has jumped around. He did an MSc in Spain, did a PhD back in Venezuela at Universidad de los Andes in Mérida. And Alberto's been a linchpin in lots of activities of the network, and partly through his work with the amazing Incubadora de la Ciencia, who are based down in Valencia as well. He's doing great things on a shoestring, but hopefully Alberto is going to give us an overview of the situation in Venezuela with a kind of diagnostic key angle on it, perhaps, but I'll leave it up to you. Thanks, Alberto.
It's been a difficult task, this one that Martin has assigned, of speaking about vector-borne diseases in the COVID times in Venezuela, but I'll try to do my best. So I usually start this talk by mentioning, if you go to Africa, you're always looking for the top 5 on your safaris. And kind of that applies also to tropical medicine. You're looking for the big 5s in tropical medicine: malaria, leishmaniasis, leprosy, trypanosomiasis, whether it's American and Africans, schisto and filarias. Turns out to be that actually Venezuela has all of these together, and the drop-down list of other neglected tropical diseases is endless. And now we got to add the viruses into the menu. So it's certainly fertile ground for research in tropical diseases. I also always mention to my students that if you're in Venezuela, you're in the Americas, you're also always looking for horses, you know? You don't look for zebras, right? But sometimes things pop out, and that was the scenario of 2020. We got a new visitor. And that was the SARS-CoV-2 pandemic hitting the Americas. So we're now approaching close to 124 million reported cases of SARS-CoV-2 worldwide, with approximately 2,200,000 reported deaths to date, if you look at the global stats in the Johns Hopkins dashboard. And we had early warned that this was going to have devastating effects in Venezuela, particularly in the context of its crippled public health system. So, we had learned about the experience in China, at what is evolving in Europe, sitting up here in New York, hits New York, and it's not until we see the real magnitude of the problem that we, look at what this is going to imply for South America and particularly Venezuela. So we rushed a note to Lancet saying that definitely Venezuela was not prepared to face this. And even though the impact of the SARS-CoV-2 pandemic has been very atypical, especially in the initial stages of the disease in Venezuela, it is starting to pick up dramatically. And we're starting to see these consequences that we had put forward on our note now, right? So I'll talk a little bit about those elements. So what's happening in Venezuela? So there's approximately 150,000 cases reported to date, with approximately 1,500 cases of deaths. But we do know that because the public health system and reporting system in the country has been dismantled, these are largely underestimated numbers of what the real situation is. Add to this also a compromised diagnostic capacity overall. So there's misdiagnosis. PCR testing is not widely distributed or available throughout the country. It restricts itself to the country. We do not have an adequate genomic surveillance system. That's also restricted to very few institutions in the capital, which are basically the Venezuelan Institute of Science, EVIC, and the National Institute of Health. And there have not been effective countermeasures to halt the pandemic. So it's starting to pick up dramatically. Information that is reliable with huge effort is that one that comes out from actually the National Academy of Science, in which one of our VenConven members actually plays a very important role, which is Maria Eugenia Grillet. So you can see in this graph, using the Imperial College of London model, how the number of confirmed cases actually does not match, not even close to the mean estimates in the range of the upper and lower estimates of what truly is happening in the country. So we wanted to look at this early in the pandemic. As I mentioned, it was a very atypical start because there was an early lockdown in the country. And there was also a situation of lack of fuel that contributed to the lockdown and a reduced mobility that influenced largely the spread and transmission of the disease. So we didn't see cases really pick up until August and October, when they made their first peak. At that point, the only way we could have access to samples from the country was actually indirectly by picking up cases from pendulum migrants that were crossing the border from Venezuela to Colombia. And that's where we were able to pick up several patients and have a snapshot of the genomic landscape of what was going down in Venezuela. We do know that the index case was a 41-year-old female traveler from Spain. So we were kind of expecting to find a genomic landscape that was very rich in the open borders, first because of the European immigration, and then because of the very porous border between Colombia and Venezuela that would mimic the same scenarios, as well as the southern border in Brazil. So the very first genomes that we were actually able to look at with our sister institution in Colombia, Symbure, led by Juan David Ramirez, who is a great collaborator. We looked at the genomes, and we found that the predominant, and that was consistent with what was happening in Colombia and elsewhere in South America, was predominantly genomes belonging to the B1 lineage. So we found 2 genomes from the B1 lineage and 1 from the B113. However, this was really early, and we were also able to pick up point mutations on the spike protein that determined the D614 G substitution in the spike plus 3 additional mutations across all genomes on the nucleocapsid gene. So these very early findings were actually very revealing of what was happening. And then we managed to expand this with further studies from Colombia. We also warned early that this was going to have impact on vulnerable populations from the Amazon basin. So we put forward also this note on profiling what the impact would be on the Amazon region, not only in Brazil and Venezuela, but also in Colombia and other Andean regions. So here in this graph, you can actually see the incidence of SARS-CoV-2 and also the lethality rates per 1,000 infections. So if you add to the math SARS-CoV-2 plus the already endemic and re-emerging vector-borne diseases in Venezuela, this is a cocktail for disaster. Let's take a quick review of the main climate niches in Venezuela. You can see that Venezuela is very rich in climate areas. You have tropical moist forests, you have savanna, you have serican deserts, you have tundra. And this basically favors the existence and endemicity of most of the vectors for all the top tropical diseases, and also to a very rich list of arthropod-borne viruses and other viruses. So here you have kind of the endemic areas with the diseases spread through the geographical landscape of the country. And adding to that, SARS-CoV-2, which has now become prevalent throughout the entire country. Also, Venconven put, this was one of the first manuscripts produced by Venconven after our meeting in Ecuador, where we put forward what the reality and the true numbers to date, despite the severe underreporting was. And we'll start with arboviruses, which have, played an important toll lately in not only in Venezuela, but throughout the continent. So if you go by the PAHO statistics, you would see that we were almost close to 2 million reported cases of arboviral diseases in 2020, with dengue being the main protagonist with close to 97.3% of the cases, followed by chikungunya and then by Zika. So in 2019, it was interesting because it was one of those epidemic peaks, endoepidemic peaks of dengue spiking and then it dropped in 2020, which with different hypothesis is that probably this was misdiagnosis or underreporting because of the lockdown. However, we do now, an increasing number of case reports are starting to pop out of co-infection of different dengue serotypes and SARS-CoV-2 in the region. So here we see this interesting graph as well, where you can see the distribution of reported dengue cases and the proportion. And even though the number of dengue cases rise exponentially, the proportion of severe dengue cases actually dropped. So I'll skip this one. This is the graph we included in our in our Lancet paper, where you can see marked by arrows the epidemic peaks in the interrenal dengue behavior. And here we have a heat map that basically states you can see all the states in Venezuela, and you can see actually how dengue was more prevalent in this heat map on the states of Amazon and Bolivar. This coincides with the area that also is facing right now the most number of SARS-CoV-2 cases at the moment. And this has to do a lot with the mining activity down south, which also overlaps with the numbers of malaria. And we'll see a paper as well of Maria Eugenia Grillet touching on that. So what's happening with arboviruses and COVID-19? So some years ago, we adopted the mnemonic of Chikdeng-Masika syndrome, kind of to narrow down the differential diagnostic list of endemic arboviruses in the region, so that dengue, chikungunya, Mayaro, Madariaga, and Zika virus that have overlapping signs and symptoms and can be confused at early stages. So is the case now. We have to add that to that list, SARS-CoV-2. And it's very difficult to distinguish both infections in early stages because of the shared symptomatology. Note also that in their pathogenesis, dengue and SARS-CoV-2 actually share the substrate of the cytokine storm and the endothelial tropism. So this obviously at early stages could have led to misdiagnosis of cases. So the question is, how had the SARS-CoV-2 pandemic wave hitting have influenced the dengue spread, right? So there are conflicted theories, and there's still lack of evidence about this. One line of thought proposed that because of the lockdowns, people will have to store water, and this will actually increase the breeding sites for the mosquito vectors, and that will lead to an increase in dengue. However, there is no solid evidence that that had happened. As Roberto actually mentioned yesterday in his superb talk, one cannot see this as a homogeneous phenomenon. And if you dig in the current literature, you would see that the behavior of dengue throughout the SARS-CoV-2 pandemic, the very scarce info that there is out there, is actually very heterogeneous, with areas that are really revealing actually a rise in cases, and also areas that are actually reporting a historical drop on the number of cases. So we'll see. But it reached the news. We don't have numbers from Caracas, but there was a lot of water shortages in Venezuela. So that led a lot to the storage of water. We should see how dengue is behaving nowadays. So focusing on the arboviruses, the important measures that should be taking is prompt diagnosis and case management, vector control and surveillance, and community involvement. None of these elements actually exist in Venezuela. So the public health system has been dismantled, as I mentioned, so there is no accurate vector control and surveillance measure, despite the few efforts that independent groups are actually doing. And for example, the efforts of the Academy of Science on building up continuous report of the SARS-CoV-2 pandemic. So there are other viruses that we know, our group and the Venezuelan Science Incubator has done very shy in the scope of our capacities, surveillance in the central western area of Venezuela. Especially throughout the Zika epidemic, that we were able to reveal concurrent epidemics of Madariaga viruses and prove also the long-term endemicity and presence of Mayaro. So we put forward this citizen science project, which is called Arboscience, which is devoted to actually perform surveillance of arboviral diseases and its vectors. And this is in tight collaboration with Maria Eugenia Grillet's group in the Institute of Tropical and Zoology. So we aim to monitor in real time the occurrence of arboviral infections, vector surveillance. We do geolocation and generate reports, and we actually do a lot of community engagement for this. So the incubator has created a real-time monitoring dashboard where you can report. This is a snapshot of the dashboard, and you can actually here see, because the pilot study was performed in the city of Barquisimeto, you can actually see that the reporting mainly is from there. And the importance of this dashboard is you can actually see the type of mosquito traps that are being used and the infusion substrate that those traps contain. So this is a very interesting pilot that's ongoing. So malaria, we've heard a lot about malaria. Professor Noya did some very important comments yesterday. Venezuela, so the America has actually witnessed an unprecedented increase in the number of malaria cases. Venezuela has been the main player in driving this increase in the Americas, with other endemic regions in the continent like Brazil, Colombia, Guyana, Nicaragua, and Panama. Most of the malaria cases are in the southern Venezuela, in the Amazon and Guyana basin, and the most vulnerable communities, of course, again, as I mentioned for SARS-CoV-2, is the Amerindian population south of the Orinoco, which are the most affected by both of these diseases. So the pandemic has actually hit hard the malaria elimination efforts in that, you know, it's basically disgregated the healthcare networks. The main focus has been, if there was some focus on malaria, this has been dropped to focus on the COVID-19 triage and contention measures. So there's been a reduction on the few malaria staff that was available to control malaria. But however, it must be mentioned that because of the COVID-19, we have actually witnessed a drop on malaria cases. Whether that has been because of lockdown measures or because of underreporting, that remains to be seen. So here you have the stats of malaria in the Americans, and you can see how Venezuela in 2019 was the main contributor of malaria cases, with almost 398,000 cases. But as I mentioned, there has been a reduction in cases in the region, and that has basically been because of a reduction of cases in Venezuela. So here's the latest paper from Maria Eugenia Grillet, in which she actually characterized the hotspots, Venezuela as being the hottest hotspot in Latin America, and this being restricted to those areas in the south where the mining activities are ongoing in context of the government implementing the Orinoco mining arc. So cases reached a historical 1 million between 2007 and 2018, with Venezuela contributing over half of these cases, 53 and 51% respectively. And most of the incidence of malaria is happening in the southern states. So here you have a graph where you can see the number of confirmed malaria cases on the solid lines. And here you have actually the maps of the southern state of Bolivar, where most of the mining activity is ongoing. And you can see the 2007-2017 progress in annual parasite incidence. I'll skip that one. And when you go and examine the occupational categories for the malaria patients, not surprising, gold miners take the lead. So it's necessary to highlight that malaria is the big player here, but other questions need to be addressed. Yellow fever, right? There's ecological transgression here to a classic niche of yellow fever. So what is happening with yellow fever? There's been unofficial reports of potential outbreaks happening down south. Also, arboviruses that circulate endemic, we don't have information about that. And, you know, it's heartbreaking to see not only the increase in these diseases, but also the unprecedented ecological damage that's being inflicted to one of the most important regions in the world in terms of biodiversity. So actually there's gold mining activities happening feverishly inside the Canaima National Park. Here you can see some of the heartbreaking pictures of what's happening down there. I have very few minutes left, and I need to run through these other vector-borne diseases. So I'll quick mention on Leishmania. The most recent stats on Leishmania report close to 50,000 new cases of cutaneous and mucocutaneous leishmaniasis, with close to 41% estimated population at risk for the disease, and close to 20% incidence per 100,000 inhabitants. 83% of these cases were diagnosed by laboratory, and there's a 66% cure rate reported in total. If you look at the intensity of reports throughout the continent, notice that actually our area where our group operates in Lara State is depicted as one of the intense regions of occurrence for leishmaniasis. And here you can see in total how the number of cases in the Americans and by sub-regions. So Barquisimeto is actually in the central western plains in Venezuela, but it also is considered the origin of the Andean mountain range, starting in Cerro Terepaima. So if we go further down to Trujillo, that's where we start building up more cases. And you would see actually here you can see the cases versus the population of risk. And here is the heat map for Leishmania as per states that we put forward on the Lancet paper. And you would see that historically, actually, the states in the central western region, like Yaracuy and Lara, where our group is located, has had the most important number of cases. By 2016, when we put this paper out, Trujillo was actually the state that was reporting the most intense burden of disease. And this is basically a state that's bordering Lara, where we actually have a group on the field operating right now, and that's Andean Piedmont. So despite that, Venezuela had a very well organized Leishmania national program that was within the scope of the National Dermatology Program that was part of the Ministry of Health, the Institute of Biomedicina, and the Central University of Venezuela. Many of these services throughout the states are not operating anymore. So we're trying to help out regionally by providing not only some sort of equipments and consumables like glass slides for diagnosis, but also members of the incubadora have been actively participating and helping out with the patients. And currently we are performing a large-scale molecular epidemiology study throughout different endemic regions in the central western areas to try to characterize the Leishmania diversity and how this could actually link to profile of disease and determine hotspots. I'll not jump to Chagas, which is our other big player. As you know, Chagas is endemic to 21 countries throughout the American, currently affecting between 6 and 10 million people. And there's an annual incidence of 30,000 new cases with an average 12,000 deaths per year. So this is a big player. We call it the trouble guy. So there's a recent update from the group of Nestor Áñez that was published in Acta Tropica, referring to the geographical distribution, and also they did the DTU characterization. It's an interesting study if you want to dig in more information about Chagas in the country. Chagas still needs a lot of work. We are working in trying to characterize vector distribution. We've been seeing some awkward behavior. For example, the classical chain of transmission in endemic areas, following the Rotnews, Triatoma, Pastrongulus, domiciliary, peridomiciliary, and sylvatic cycles is not quite happening. So we've seen an increasing trend of Pastrangilis positioning itself as the main intradomiciliary vector in areas where it was not used to be the main vector. So we are looking also forward to start working on defining some genomic landscape of the triadermin population in Lara. So why do we call it the troublemaker? And this is basically the disease where we are mainly focusing right now, is because of its insidious clinical nature and chronic behavior, and because there is a huge gap in knowledge in many communities. And now you can add the increasing oral outbreaks that we've seen in Venezuela, and we have Dr. Belkis Noya, who's become a world top-notch authority on this. We try to perform diagnosis in several endemic and hyperendemic areas of central western Venezuela. We've initiated entomological campaigns and trying to raise community awareness through citizen science project. So we've also launched as part of Venconven, we've also launched a campaign, a project that's called Trae tu Chipo, Bring Your Kissing Bug, that also has a real-time monitoring system for finding kissing bugs. So this is the way it works. Citizen sciences in the community actually report on the finding of triadermin bugs. They report it through the survey. We're trying to get smartphones in each of these communities so we can strengthen the reporting. That comes into the cloud. We immediately start the geolocalization process for this and arrange for specimen delivery into the lab so we can further do the screening for positive parasitemia on the kissing bug and alert the community whether the kissing bug was positive or negative, and then arrange a visit to the community for an extended and broader capture of specimens. We are hoping to accompany this by massive sero screening of those communities that are reporting the most cases of kissing bugs. And we manage that in an operational dashboard, which I would show you. So here's how it works. Here's the operational dashboard, citizen science report. And for here, we've had actually a more extended reporting throughout the country. We have cases reported from the south, from Bolivar. We have cases from eastern Venezuela and from the central Venezuela. We've actually partnered with the Institute of Agricultural Zoology, which has a superb team of entomologists, and our own work in central western Venezuela. So we're hoping to do a major projection of these tools and make them more available to the community very soon. So once we recover, we arrange the recovery of the specimens. Those specimens are assessed in our entomological lab to detect the parasite. And if the specimens are positive, so we report to the community the positivity. So we do not only the GI tract dissection, we also spot the feces onto filter papers for molecular characterization of the parasite. So here you can see actually the moment we see the parasite in the fresh prep is the moment where we actually report to the community the positivity and start arranging for massive community retrieval in the communities. And then we go on to molecular characterization of the parasite in the feces. So these are community outreach and citizen science projects. We believe that education is the backbone for everything. And what we've achieved so far is thanks to our citizen scientists. So Venconven has actually played a very important role in the launching of these of Arboscience and Bring Your Bug, we are now actually working more intimately with other people on the network to develop and come up with more public engagement and public outreach programs. And as I mentioned earlier, one of the major collaborators of Ben has been our sister institution in Universidad de Rosario in Bogotá, Simposio, led by Juan David Ramirez. And currently members of the Venezuelan Science Incubator are actually performing training in molecular diagnostics of endemic diseases that we are planning to bring to Venezuela to continue these projects. So,
Greg Abel is an arbovirologist. He's currently based at Colorado State University. He's got interests in arboviral diseases, virus-host interactions, arboviral surveillance. He's worked at New York State. He's worked at the University of New Mexico. And he's pioneered this approach called xenosurveillance, which is really exciting. I'm going to stop waffling on. We're just very excited to have you here, Greg.
Yeah, thanks so much for inviting me. I've been kind of lurking on and off during the last couple of days, and it's been really interesting to kind of hear the other talks. It sounds like a lot of us are kind of grappling with the same issues and kind of trying to make the same kind of progress. So what I've got for you this morning, let's see if I can figure out how to make mine. There we go. I want to just kind of introduce you to xenosurveillance as a concept and give you a bit of background on why we got interested in it and give you a little bit of the history of the project. It's been going on in my lab for quite a few years now, kind of limping along, and it's one of my favorite things that we've been doing, and we are finally kind of starting to make some headway with some significant funding on it. So it's kind of an exciting time for the project. I'm going to give you a bit of background on kind of the methods and the approaches, and kind of walk you through some of the data on why we think it's useful and a potentially quite helpful tool in certain types of situations and environments. And then just give you a bit of information on what we're doing now. And this is very much a work in progress, and it's becoming clear the areas where we need to do a lot of work. And so that's kind of what my plan is. But where I want to begin, which I think won't be news to any of you, is this idea that we're living in a post-continental world of infectious diseases, where the kind of globe that we look at when we look at a map of the world is sort of with respect to infectious diseases, kind of a figment of your imagination. And I think the fact that this meeting is happening online and that none of us have seen each other in person for the last year is testament to that. And the world that we live in, I think, looks a lot more like this one from 220 million years ago than the one that we think we live in. And so we've done a really good job over the years of globalizing viruses and other pathogenic organisms. And in the world of arbovirology, this goes back to the slave trade with the global movement of yellow fever virus and the mosquitoes that transmitted Aedes aegypti from Africa to the rest of the world. Yellow fever is thought to have originated in a small focus in animals in Africa and spread throughout the world as a result of the slave trade. Similar thing happened in the Great Wars of the last century when dengue virus was also globalized, and we now are in a situation where dengue is a global pathogen, quite successful. This happened again famously in 1999 when West Nile virus was introduced into New York City. And at the time, this was seen as a shock, but really it shouldn't have been seen as a shock, because prior to its introduction into North America, there had been these little outbreaks outside of the known focus of West Nile virus in the Rift Valley region of Kenya. So there had been outbreaks in the Middle East and in Russia and the Volga Delta, small outbreaks, and then there was this explosive epidemic that now sort of is all over North and South America. Didn't have to wait long for this to happen again, and if you think about what happened when chikungunya virus emerged, it's a very similar picture, right? So there was this initial focus of transmission that had been known historically in West Africa. And then we started to see little outbreaks here and there, followed by the sort of florid explosion of chikungunya, which is now sort of transmitted throughout the world's tropics. And again, this happened most recently in the arbovirus world with Zika virus, and then shortly thereafter with SARS-CoV-2. So this is a pattern now, and I think that what we're going to be facing as we move forward is situations where this is becoming more and more common. So there are a lot of reasons that this is happening, and again, in the arbovirus world, we tend to think about globalization, which I've already spoken about, the rise of tropical megacities and urbanization of the tropics. That's unprecedented. There are many, many, many more humans living on the planet than have ever lived there before. And we also have done a great job of creating environments for certain mosquitoes, which have adapted as they will to what we provide them. And so I'm thinking specifically about Aedes aegypti and Culex fasciatus and related mosquitoes. So, this emergence of new pathogens is happening at a lot of different scales, right? So there's virus adaptation to mosquitoes and mosquitoes that interact in different environments. Can anybody see my, I don't think my pointer's showing up there. Maybe, can you see my pointer?
Yeah, we can. Yeah,
okay.
It's white on white, but we can just about see it.
Yeah, I don't really know how to fix that on Zoom, but that's okay. Doesn't matter. Yeah. So anyway, the point is just that this process of emergence is happening on a lot of scales, and none of the factors that I talked about seems likely to me to kind of get better anytime soon. And so what we really need to think about is, how are we going to adapt to this, and how are we going to do a better job of detecting viruses as they emerge and monitoring them once they do so. So we spent some time many years ago thinking about when we would want to look at this. And if you just look at this line here, this is kind of the what we think of as an epidemic. And so there's a few phases of this. Early on, we think about a new virus as a zoonosis, right? So this is something that's not really going to affect people. And if you think about SARS-CoV-2, that's a great example, right? So not that long ago, that virus existed only as a pathogen of bats or whatever. And then as time goes on, you get these initial little spillover events where the virus passes back and forth, and maybe it establishes itself, but maybe it doesn't. But then at some point, the virus becomes, or the pathogen, we don't have to be a virus, it really emerges in some kind of new host, and that opens up possibilities for this epidemic to occur. Okay, so when can you detect that? So if you're a virus hunter and your approach is to go out into the world and sequence everything and just collect a bunch of stuff and see what's there, you could detect that at that very early stage of this kind of zoonotic event. If you rely on looking at like Twitter and news feeds and Facebook, as many do, to detect these things, you can kind of detect it later on, but you've missed this really critical area where the virus has already emerged into a new host, potentially humans, but we don't have an outbreak yet. And that's kind of when you'd really, really like to be able to know about it. During the epidemic, however, there also really is a need in resource-poor environments for enhanced monitoring and better ways of detecting a pathogen as it's spreading and moving into new ecological niches so that we can monitor who's getting infected, what species are getting infected, is the virus evolving, and so forth. There's also, I think, a role for enhanced surveillance in post-outbreak monitoring, because in the case of Ebola, for example, you'd like to maybe know whether somebody who had been infected was going to recrudesce, or whether the virus is going to emerge in a place where we already knew that it had emerged months before. So this is that's kind of the problem that we're facing. And so we first started discussing this idea of xenosurveillance in our lab at around 2011, and the reason that we did so was that kind of a lot of factors came together. The first is that my colleague Brian Foy, who's up here, was working in Africa collecting dried blood spots and blood spots from mosquitoes as a part of an ivermectin study of malaria. Brian is famous on the internet for inadvertently discovering that Zika virus can be transmitted sexually as a result of coming home from this from this field trip, and he's my neighbor, and his wife was certainly not very happy about this discovery that they made together. So he was doing that work in Africa. Next generation sequencing was coming into wider use and becoming more accessible. This is something that we had done in the lab, but we were kind of starting to think more about how we could use it. Computational approaches to the large amounts of data that one gets through next generation sequencing were becoming more accessible to labs like mine, which aren't principally computational labs. And then also there were some hints in the literature that arthropods would take up non-arthropod-borne agents during blood feeding. And I had just moved to CSU along with a really great postdoc, Doug Brackney, who's pictured here, who got really interested in this idea. And I was looking for ways to collaborate with my new colleagues. And so that's kind of how the project got started. The core concept here is that in resource-poor environments, such as Africa, such as Central America, parts of South America, mosquitoes are out there and they're feeding on people all the time anyway. And that in the blood that they're taking from people is information. And if we can access that information, then we can learn a lot about the health of the people who these mosquitoes are feeding on. What we didn't realize at the time was that they're also feeding on lots of other animals that are around. And so we can also learn about the ways that pathogens are moving back and forth between wildlife, domestic animals, and people. Another key piece of all of this is that a mosquito comes and takes a blood meal on you, and then it's big and fat and full of blood and it can't fly very well. So what it does is it goes over to the wall of the dwelling or where some convenient close place, and it sits there. And that's a vulnerable time for the mosquito, and they're really easy to go and collect. And I'll show you some pictures of how we do that in a few minutes. And yeah, so we just had this idea that blood meals contain information on mosquito behavior and mosquito susceptibility to insecticides and the exposure histories of what they're feeding on, and also, you know, pathogens, including non-vector-borne pathogens. So we got kind of excited about this and started kind of trying to think about it more. And this is kind of the, you know, our dream of how we drew it up, how it might work. So this is a dwelling in Central America, and you know, we go in and we use these things that are called insectizookas to slurp blood-fed mosquitoes from around the inside of the house. We bring them to the lab, we anesthetize them, identify them, express the blood meal. Here's a blood meal in this mosquito. Express it onto a preservative card. This is a FTA card. And then those can be stored, then sent back to the lab where we can process them by punching out the blood, extract RNA from the blood meal, and then either test it by PCR or sequencing, whether it's an Illumina or an ONT type approach. And then we get data from that, and that can tell us something about what type of intervention might be done or what kind of pathogens may be burdening the people who we're working with. So that's how we thought it might work. But really, we don't, that's sort of just a nice idea. We don't really know whether it would actually work. And so we set out to answer the question, could this ever work? So the first experiment that we did was to ask whether viral RNA could be detected in mosquito-derived dried blood spots. That's what the MDBS thing stands for, by qRT-PCR under laboratory conditions. We designed an experiment where we took mosquitoes, Anopheles gambiae that had been reared in the lab, and we fed them either on an artificial blood meal or on a hamster that was infected with either West Nile virus or HIV-1. We then held them either for 12 hours as kind of a standard, or for a time course of like 30 minutes, 6 hours, 12 hours, 18 hours, or 24 hours. We then took the mosquitoes, squashed the blood meals onto these paper cards and that preserve RNA and DNA, and then stored them at room temperature for 2 weeks, and then did this processing where we extracted RNA and then tested them by qRT-PCR. And so these are the results of that first experiment. And this was done by a really good grad student in the lab, Nathan Grubaugh, again, many years ago. And what he found is that if you just take blood and extract RNA from it, and then take that same blood and put it on a FTA card and extract RNA, you lose a bit of sensitivity by doing that. What you don't lose, you do not lose further sensitivity, however, when you let the blood sit in mosquitoes for 12 hours. So this is just directly 2 microliters of blood on a dried blood spot, and then a mosquito dried blood spot, and that's panel A. I can't get my pointer to work anymore. Then we did also a time course, and what you kind of see from that is that you do start to lose sensitivity after about 12 hours for both of these, more so for West Nile virus. And remember, West Nile virus is not transmitted by Anopheles gambiae mosquitoes. They're not competent for the virus. But we're able to detect HIV out to 12, 18, and 24 hours in all of the mosquitoes that fed on that blood meal. So this is encouraging for a couple of viruses. We then became curious about whether we would be able to detect a broader range of viruses by this method, and then also whether we could detect bacterial and parasitic pathogens. And so the next study that we designed was to assess that. That was done by another grad student in the lab, Joseph Favre, who you can see there. And the experimental design is again over on the left of the slide. And it's very similar to what I already told you about. We feed the mosquitoes either on one dilution and then do a time series, or on a serial dilution of the pathogen. These are serial 10-fold dilutions, and then process the samples for xenosurveillance as we do it. And so the pathogens that we look at in this case, also not transmitted by Anopheles gambiae, are Bacillus anthracis, that was the Stern strain, so we're not working with anthrax in the lab. Trypanosoma brucei, Zika virus, and MERS coronavirus. And so what you're looking at in this set of figures is this dilution series. So we took the different pathogens, made serial 10-fold dilutions, fed them on mosquitoes, and then processed them. The shaded part of each of these panels is the clinically kind of observed range of viral loads. And so the question is, can we detect this at or below clinically relevant amounts of virus? And so what you can see from this is that yeah, we're readily able to detect this in some cases down to very small amounts of each pathogen in the blood. This is after 12 hours. And then this is a time series, like I showed you before, with each of these things. And the reason that we do this time series is that we're imagining that in the field, mosquitoes are feeding on us or on the people, and they're sitting in the dwelling. And so how fast do we have to get there is kind of the question that we're trying to answer here. And so, do we have to get there at 4 in the morning within 6 hours, or is it okay to get there at 10 in the morning or later in the afternoon, and will we still be able to detect these? And so the answer from all of this is that this seems to be a fairly robust way of detecting pathogens in the blood. And so yeah, that's just the conclusion. Clinically relevant amounts of pathogens can be detected in mosquito-dried blood spots for up to 24 hours post-blood feeding. And this data that I'm showing you is for Anopheles gambiae. We've now done similar experiments with Aedes aegypti and Culex fasciatus, and the performance of the approach is quite similar regardless of what mosquito is. Aegypti and Quinks are more relevant to Venezuela, certainly, than Anopheles gambiae would be. Okay, so that all is done by QRT-PCR. We then decided that what we really would like to do is, we don't think that we're smart enough to know what we should be interested in, and certainly in the case of a virus or a pathogen that nobody's seen before, we wouldn't be able to detect it if we use these really targeted primers and probes. So we tried to do next generation sequencing, and this is the preliminary results, and I won't walk you through it except just to say that it made us very sad because there was really no pathogen signature in this whatsoever. And so what became really clear was that we needed to develop better methods for doing our sequencing. So we then set out to do that, and we took really two approaches. One was to use baits to pull out sequences of interest. The other was to deplete RNA that we don't care about. And what I'm sort of skipping many years of labor and frustration, and just telling you that what we're now doing is we're not doing any target enrichment. We really are just depleting ribosomal sequences. And there's a paper here that describes, it's the Favre et al. 2019 paper from Virology that describes exactly how we do that. The problem with our input RNA is that it's loaded with mosquito ribosomal sequences that we don't care about. And so what we did was design a series of probes, primers actually, against all of the all of the mosquito ribosomal sequences that we thought we would encounter, and then use an RNase-H-based approach to selectively deplete those sequences. So the way that works is shown in this kind of figure over here. So we have this mixed input RNA. We add our probes to the ribosomal sequences and then amplify based on those just in a single strand, so that you have RNA DNA hybrids, which is the substrate for RNase-H. So you can then selectively degrade all of that stuff and then DNase treat everything. And what you're left with in the end is just non-ribosomal RNA, which is what we really care about. And again, what you can see a bit on the panel to the right is that this vastly improves our NGS performance. And I would be quite happy to talk in detail about this with anybody, but I wasn't sure for this audience whether you wanted like a real detailed explanation of how that works. But I would highly recommend for NGS for mosquitoes that you undertake such an approach. Okay, so that's now great. Many years of work in the lab trying to figure out whether our protocols would work. We also then, who cares if you can't really do anything with it? And so the first way that we tried to do this was using some field sites where our colleague Brian Foy, who I talked to you about earlier, was doing this malaria study. And this was in Lofa County, Liberia, which many of you know is where Ebola virus emerged. And so we were trying to do this in the context of a sort of waning Ebola virus outbreak. So this was a cross-sectional study that was very fast, very dirty, done over the course of 2 weeks in 2 villages in Lofa County. In one study, we just went and collected mosquitoes. Another part of the study involved using visiting houses, collecting mosquitoes, collecting capillary blood from people who slept in that house the night before, and then we assayed these samples by next generation sequencing, PCR-based analyses. And this table here, okay, so these are the guys who help us collect mosquitoes. They're our important partners with the Liberian Institute for Biomedical Research. And then this was the field team over here, Nathan, James Wager Lucarelli, who was a postdoc in the lab for a number of years, and then Joseph Favre, looking a lot less clean cut than he was in the last picture. So we collected several hundred blood-fed mosquitoes and made pools of them for sequencing, and I'll tell you a little bit about what we found when we did that. This was this kind of more convenient sample of mosquito-derived blood spots only, and what we were not particularly surprised, to be honest, to find was a lot of Epstein-Barr virus sequences in these mosquito-derived blood spots, depending, not really dependent on whether it came from village A. Village A, day 1 or 2, day 3, day 4, day 5, and then village B. We found a lot of Epstein-Barr viruses, which is something you'd expect in Africa. We were also really interested because we found a mosquito pool that had a signature of canine distemper virus. And so that's kind of interesting because that's a virus that seems to have this ability to cross species a lot. And finding that in a blood meal of Anopheles gambiae that probably fed on a person was sort of kind of jarring. So we went back to the sample. We were able to re-extract RNA from the actual blood spot that yielded those sequences and identified that the blood meal was taken from a dog. So this was one of those examples where Anopheles gambiae doesn't feed on people. It feeds on sort of whatever is there. So the more important piece of data from this, in my view, though, is that we were able to detect a couple of viruses, both in mosquito-derived blood spots and in human-derived blood spots, and that was GB virus C, also not highly surprising given where we were collecting this, and hepatitis B virus. So in this case, the blue is sequences that came from people, and black is from dried blood spots from mosquitoes. And so this kind of, in our opinions, kind of proved the principle that pathogens sampled from mosquito-derived blood spots reflect those that were present in humans. And this is quite encouraging, in fact. And so this has been one of the pieces of data that's kind of kept us pushing on this project for as long as we have. Another thing that's really interesting is that when you start to analyze this data, you learn a lot about the virome and microbiome of mosquitoes and people that you're sampling. And so I just want to show this because one of our really important collaborators has been a computational biologist, Mark Stenglein, who's a colleague also at CSU. And so part of this, one of the benefits of this project is that we were able to discover this really, really divergent Anopheles flavivirus. And at this point, you know, Anopheles don't transmit any flaviviruses. And so to find a insect-specific flavivirus in Anopheles was really interesting to us. And so I just show the data to point out that, you know, you never quite know what you're going to get when you go and start sampling things from this type of environment. And we think that we are kind of also, you know, contributing to basic knowledge about the evolutionary history of virus, mosquito interactions, and so forth. So I won't dwell on that, but I just wanted to point it out. So this is all, you know, in our view, really encouraging. But what we really would like to be able to accomplish with this project and with our work is to, you know, really have an impact. And given the extreme need for better ways of monitoring infectious agents and the kind of really big problems that we have in the world, we're starting to kind of ask ourselves, okay, well, you know, can we kind of transform the way that we think about and conduct surveillance given this level of problem? And so what you're looking at here is a backyard of a house in rural Guatemala. And this is from the story that the part of the project that I'm going to tell you about next. And it's quite typical of this area, and I'm sure this won't be surprising to you. Here's you know, there's some pigs in the backyard, and there are ducks that you don't quite see, and there's you know, some chickens walking around, and this is the latrine, and there's a pile of tires and so forth back here that are loaded with Aedes aegypti mosquitoes. This is the rainy season, so this sort of puddles everywhere. And that's really, really typical. And so in this kind of environment, you know, how do we, you know, can we kind of change how we do this? And we think that the answer is yes, but that doing that means that we're not going to kind of be able to do things the way that we've been doing them. And so what does that mean? So the first thing that we're really trying to do is to kind of take this One Health approach to heart. And so what we've done over the last couple of years is to, you know, redesign our team. So we have, you know, this isn't just like a Greg project. Here's me, and I'm kind of a virologist and sometimes an entomologist. But we have an infectious disease doctor, Dan Olson here. We've recruited veterinarian, Valeria Scorza, and we're kind of trying to bring, you know, the expertise of these different people in our institutions in Colorado into it. And it really has, I mean, I'll just kind of say that the proposals have become way more interesting and a lot better that it's now that it's not just kind of me thinking about entomology and virology, but that we have people who kind of know what they're doing in these other areas. What Dan in particular also has to contribute is that he works at this really well-established clinical research site in rural Guatemala. And you can see it's in this region here, about an hour outside of Coatepeque, near the border with Chiapas. And here's a picture of it here. It's actually a quite well equipped research lab, and there's places to stay, which you can see here. So you can actually go there and get a lot of work done. And so the other thing that we're doing is, like, if you think about the pictures that I showed you recently in the previous slides, you know, you can't just think about what are the mosquitoes and what's in the mosquitoes. You kind of need to be thinking about, you know, how is it that the animals and the people are interacting. And so as the project grows, we'll need to bring in the social sciences and so forth. But we haven't done that yet. The other great thing about this site is that there are really, really, really good local trained people who are there, who are from the community and who are in the community and can do this work in ways that I could not. And even Dan, who's worked there for many years, cannot do that. So that's kind of the first thing that we're trying to do. And we just recently have a grant to well, the grant started about a year ago and immediately was put on ice because we were not allowed to travel. But unfortunately, the clock keeps ticking on the grant, so we're trying really hard to get the project started. But the overall aim of this is to compare a syndromic approach where you go and you ask people how they feel with xenosurveillance, where you kind of go and collect mosquitoes and see what's in them. So we're in the process of enrolling 40 households in 2 villages in the Ultrafinio region. At enrollment, we're collecting capillary blood from humans and up to 5 animals, and it's chickens and dogs and ducks and pigs. Occasionally there's larger animals there too. We aspirate mosquitoes from within the house. They get a health questionnaire, which they then get every week. The study nurses visit each of these houses once a week. Once the mosquitoes get back to the lab, we sort of do this xenosurveillance approach. They're identified. We express the blood onto FTA cards, and then the study is going to last for a year. And it's going to be really interesting to see what we learn when we do that. We began enrollment just a couple of weeks ago, and so stay tuned. We don't really know how this is going to turn out, but we're certainly very excited to see where it goes. The other thing that we really want to do to make this project be transformative is to get away from this way that we've been working, where we go and we visit a site and we collect the stuff and then we bring it back to CSU, and it takes us many months to kind of get that done because we're involved in lots of other things. What we really want to be able to do is to use local lab capacity much more effectively. And you know, we've been inspired by a lot of these projects, some of which we're just kind of heard about in the last hour or so. So we're kind of trying to emulate that. And that's, you know, I'd love to say that was going to be straightforward, but I don't think that it's going to. You know, this is not an easy project to do. And so to the extent that we can help shoulder the burden here in the North, we'd like to do that. And that's also the way that we do things. Also, you know, moving things from here to there is expensive, and this isn't yet. We don't have a ton of money to do this. I see a barrier also in training. Some of the protocols are technically challenging, and then there's this issue of how do we analyze data? Because what we're envisioning being able to do ultimately is to do NGS and QRT-PCR on site. That's our goal. I think we can get there. All of these are surmountable. But I think it's important just to try to, you know, identify what the barriers are to getting where we want to go with the project. And then so this is the pictures down here are the people whose job it's going to be for the next year to try to make this happen. It's Emma Harris, who's a postdoc in the lab, and then Karen Arias, who is a lab technician in the field site in Guatemala. So I'm going to slow down here. Hopefully in the last, I don't know how long I've been talking, probably longer than I should have. I've convinced you that we really, really need new ways of doing surveillance. And hopefully I've also convinced you that xenosurveillance is technically and analytically feasible, minimally invasive. You know, the one thing that I didn't mention earlier is that going into somebody's house and asking to collect mosquitoes gives you a totally different interaction than going into somebody's house and asking to take blood from their kids. And so, again, we're not there yet because we're still at this comparison stage, but what we envision is a project where, you know, we're really convinced that what we can get from mosquitoes reflects what we would get if we had gone and bled everybody in the house. Yeah, hopefully also, you know, you can see that there's a lot of new information that one could get by doing this. Pathogens in people, pathogens in mosquitoes, microbiome, trying to learn more also about the mosquito animal human interaction. One thing that we're not able really to do very effectively with this study at this site is to monitor wildlife, domestic wildlife and domestic animals and people, just because we're not sampling in that space very well yet, but I think that also would be really important. I just don't know that we're in the right place to do it. And we have a lot of work left to do. Like I sort of indicated earlier, we'd like for this to be a transformative project, but we don't really know what the right way to use the sample is yet. We have some ideas, but again, data will sort of rule the day when we have some. And then we also have this big challenge of trying to migrate expertise to where it's needed the most, so that it's not so much about us, but more about empowering people where they are. So there's so many ways, so many people who need to be acknowledged. Most importantly, the people who are participating in the study and who are agreeing to let us into their houses. We've gotten bits of funding over the year from a lot of different sources. We're currently supported by the CSU One Health Institute and by the US National Institutes of Health. And then there's a list of people there. It's been sort of one of these projects that's gone on in the lab for a long time, and it's never really been anyone's sole project. It's been something that they've done because they like it and they were interested in it. So I think that I will stop there, and if there are questions, I'll do my best to answer them.